Hi students, this is Mr. Eckert. We're going to go through target H5, which is probably going to be a little bit of a review target because I think we've talked about these concepts before already in class, but the target says you should be able to explain the significance of introns and exons in DNA in terms of their gene expression. So I think you'll remember that introns and exons are both parts of the gene in a DNA. So a gene, again, is a segment of DNA that codes for a particular trait or protein. The exons are the actual coding portions of that gene, whereas introns are non-coding portions. So again, I think I may have shared with you already, the way that I remember this is I think of the introns as interruptions in the coding sequence, and exons is actually the parts of the sequence that are expressed into proteins. So introns, as it shows here in this diagram and explains, can range in size from 20 to 30 bases to thousands of base pairs. You can see different segments of this gene are labeled as exons and introns. And again, I think I pointed this out to you already in class, but just by looking at the DNA double helix itself, it's impossible to tell which segments are introns and which are exons. But in any given gene, there you're going to find both exons and introns. Now, here's something that is probably new. Introns, when the initial RNA, messenger RNA, is made from the DNA, the introns are actually present. That's known as pre-mRNA. Then there's an enzyme that comes along and snips those out, or splices the, takes the introns out and splices the exons together, and that creates what's called mature mRNA. So mature mRNA is made only of exons, whereas pre-mRNA would still have the introns, the non-coding segments, in it. You can see how it's very, very important those introns be removed in a precise manner, because if the enzyme messes up and there's introns that are left in there, or if it deletes accidentally an exon, then it can result in a mutation. The protein either may not be produced at all, or a protein that is not fully functional may be produced. So as it says here, imprecise intron removal could result in frameshift mutations. The genetic code is going to be read incorrectly by the ribosome, and once again the protein either is not going to be made at all or made incorrectly. So this is just a diagram showing DNA consisting of both introns and exons, and again, as those introns are removed and the exons spliced together, you get mature mRNA down here. So that's it for that target. If you have any questions, please make sure you ask.